Okay, so here we are now, kind of taking what we learned in the last chapter and bringing it forward a little bit more specific to specific kinds of environments and the animals we find within them. Starting as life did, I guess, in the aquatic marine realm, depending on where we're at, we have different challenges, right? So if we live in, I'm just going to pull up a white screen here, if I can do that. If we live in freshwater, freshwater, still green, like that, freshwater, our challenge is that we are saltier than the world around us, right? So, animal salty. And as a result, our problem is twofold. We are constantly gaining water from the environment. We are constantly losing, in particular, sodium, calcium, stuff, right? And so our issue here is that our internal bodily fluids are at risk of becoming more dilute. Okay. So the way that we're going to solve this problem is two ways. Okay. We are going to, so solution here for our freshwater vertebrates, we're going to constantly urinate. We are peeing 24 hours a day, 7 days a week. Okay. And by peeing constantly, this solves all water problem. Okay. But we still have another issue here. We still have to deal with the loss of all of these solutes that come into that. Okay. So the way that we're going to do that is at the gill. So just as we talked about the gill being an important mechanism for the exchange of gases, here we're also going to use this gill as a means to reclaim some of the ions that we're losing through diffusion. Okay, so we do this with two different mechanisms. One, okay, here we're going to gain sodium by exchanging it with free hydrogen ions. Okay, so it's a positive for a positive. It is electroneutral. We're also going to have another electron-neutral exchange reaction, okay, by exchanging chloride for bicarbonate. Okay. Now, where do we get bicarbonate and hydrogen? You might remember this. Hopefully, you remember this from the last chapter, or not last, the chapter before the last chapter, right? From metabolism, right? As we take carbon dioxide and we put it into your blood, we yield, that should be, sorry, equals, it should be a yield. We're going to yield carbonic acid, which is reversibly exchangeable as hydrogen atoms bicarbonate. So this is stuff we got to get rid of, right? So we're doing two things. We're getting rid of metabolic waste products, and we're also using that exchange reaction there to bring in some of these atoms, these ions that we needed from before. So we think about these guys here. These are fluoride cells, right? These happy little guys, and they exist on the gill. Okay, so just to kind of give you a sense what they look like here, we've got a Electroflora fluorescence here with some labeled ion transport proteins. They're not all chloride cells, but we just kind of collectively refer to them as chloride cells. They're also involved in the exchange of other ions, in particular calcium being exchanged here. One thing that is interesting about these ion exchange proteins is that they're big. They take up a lot of space on these otherwise relatively thin Gill lamellae, right? And remember, we've talked about the thing with gas exchange is that for diffusion to work, the membranes have to be relatively thin. So there's a balancing act here between exchanging ions, right, and trying to exchange gases as well. Okay. Where this comes into play is when we take freshwater fish and we start really altering their water chemistry, right? So if we put in fish, for example, I have no idea what kind of fish this is, but we put it in soft water, right? You may have a water softener in your house. It's something that removes calcium from your drinking water, from your laundry water, from basically the water coming in your house. And so soft water simply means that water is devoid of calcium. If we put a fish in soft water, the problem here is that it has to kind of work extra hard to pull that calcium back in, right? Because it's really losing calcium. The gradient is so big that it's losing calcium in a very fast rate, okay? So we end up with all of these additional ion exchange proteins all over these gills. And what that means is that this fish basically can't breathe because all of the gill space is given over to trying to reclaim calcium and not for gas exchange. So that can kind of suck. As soon as we get into salt water, we end up with two very, very different scenarios. Here we have a vertebrate, right? So our marine fish. And most marine fish are of this sort. They're teleosts. If you've taken comparative, you know what a teleost is. The most common bony fish most common fish on the planet. And these guys have a freshwater origin. Okay, so even though life started in the oceans and fish began in the oceans and all of this wonderful stuff, the most common marine fish that we have are actually secondarily marine. They came to them from freshwater. So they have a origin story that doesn't normally deal with salt water. So they are very much type osmotic. Whoa, there's my S. Osmotic to salt water. They are very much less salty than the water they live in. So that's a problem, right? They are constantly losing water and gaining salts. Okay, so the regulatory mechanisms are pretty intense. Our marine invertebrates, on the other hand, have an origin in salt water. So they have lived in salt water forever, and they're there forever. So they're adapted to it, right? And so they actually are essentially, they're not exactly, but they are essentially, come on, ice cause the salt water. So if you eat a squid, I would encourage you not to eat squid and octopus. They're extremely intelligent organisms. It's kind of a crime, almost a crime to eat them. But if you were to eat one, right, you essentially are getting something that is as salty as salt water because their tissues are salt water, basically. Whereas if you eat a marine fish, these tissues are going to be significantly less salty than the water around them. So these guys don't have to worry a whole lot. They just exist. But these guys, because they maintain a separate environment internally from the external environment, there's a lot of exchanges that have to happen here to make this work. They can't do this, right, in a simple way, right? So again, I'll pull up a white screen here. Right, the problem with our salt water is that they are constantly losing water and gaining salt. Our freshwater fish dealt with their water problem by peeing all the time. Okay, And that works great for them. But here the issue is that we're losing water all the time. And so our number one thing with urination is that we're going to try to minimize water loss that way. Problem, okay, is that our fish kidney cannot produce hyperosmotic urine. Our UP ratio is always less than 1. So we're always losing water from urine. Kidneys aren't going to solve our problem here. Okay. How are we going to solve any of this stuff? Well, again, you think about chloride cells. Okay. So just like before, right, we're exchanging chloride, right? Only now, the way this is going to work, okay, is we're going to do it in reverse. We are transporting chloride that we're passively gaining from the environment around us. We're kicking that shit out. It's leaving. And as that chloride gets pumped out, right, we create a negatively charged external environment. We have an electrical gradient. And that electrical gradient is actually strong enough to pull sodium out with it. So by pumping chloride out, it costs us energy, right? This is an ATP-rich reaction here. But by moving one thing, we get all these other things that we don't want that we're passively gaining from the environment. We kick them out as well. So that's going to help at least a little bit solve our problem. So to kind of sum up here the difference between our fish, right? For our freshwater fish, right, we're gaining water, we're losing salts, we're peeing constantly, and we're importing salts at the gill. And so we're bringing salt in, we're losing water this way. For our fish, we're constantly gaining salts, but we're exporting them at the gills as best as we possibly can. Okay, we're also dumping salt out in the urine a little bit. It's not a massive amount, but a little bit there. Where are we gaining water? The one thing we haven't talked about, saltwater fish gain water from food. Okay, food, and they also drink salt water. Because they have this additional little mechanism to get rid of these salts, these chloride cells, they can do so without dehydrating, right? You can't do this, right? Because you don't have any chloride cells. If you continue to drink salt water, you are very, very quickly going to die. So how does it work with vertebrates that aren't fish, but are still exposed to these high levels of salinity? That maybe their only water source, or preformed water source, is salt water. How are they going to do that? Well, they're going to do that in the same way that fish do to some extent, right? In the sense that they're going to have special glands or cells that concentrate and excrete salt from their bodily fluids, right? These are salt. Ooh, here's my proximity. Salt glands. Okay, and these are salt glands. These glands that sit usually above the eye, right? So there. Okay, and as blood passes through them, they just actively pump salt into it, right? Remember these animals, they're drinking seawater, they're eating a lot of times they're eating marine invertebrates, so they have a lot of salt in the food they eat as well. They have to get rid of it somehow. So it sits and collects in these salt glands and drains out a little duct, a little nasolacrimal duct. Okay, it comes out, I'll write that out for you if you want to know. That nasolacrimal duct comes out and it allows them to drain a very, very concentrated saltwater solution. It kind of makes them look like tears, or like they're crying. Okay, they're not. They're just getting rid of this excess salt from their diet. The other big, fun group of marine vertebrates are the chondrichthyans, the sharks, skates, the rays, chimerae. And these guys have a very, very different solution to their problem. And if you remember from the very end of the last chapter, we talked about the idea that osmotic pressure doesn't care about what, it just cares about how much, right? And so a cell might create a bunch of extra little proteins and stuff its insides with them to be isosmotic to the solution around them. Sharks are going to do basically the same thing, okay? They have the same problems as our marine teleosts did. They're constantly losing water to the environment and gaining salts. So what they're going to do is they're going to utilize a waste product, okay, and that is ammonia, from the breakdown of proteins. What they are going to do is they're going to take ammonia, which is extremely toxic, right? Please don't go drinking bleach, regardless of what you might have been told by our president. Don't do this, okay, because it will kill you. So you can't store it in your body. So most animals that leave it at ammonia have to pee constantly, right? But what we can do is convert ammonia to urea. Is what you and I do, and that's also what sharks do. And urea is extremely stable. It won't kill you, it won't do anything bad to you, you can store it in the long term. So our sharks produce a ton of urea, and then they just leave it inside their body. And so they are hypoionic, the salt water, right? So the outside is saltier than they are, but they are actually hyperosmotic. Their fluids are more concentrated than the salt water. It just happens to be that what they're concentrated with is urea and not salt. So sharks actually gain water by diffusion from salt water. Right? They don't lose water in the same way that bony fish do, because they have so much urea kicking around in their tissues. Otherwise, sharks kind of taste not great all the time. So that's one pretty elegant and fun, I guess, solution to the problem. The other solution they've got to the problem is, again, if you've taken comparative, one of the things we looked at here is at the very tail end here, near the cloaca, coming at the back end of the digestive system, there's a little finger-like thing here called the rectal gland. And that rectal gland concentrates salts, okay? So kind of like our salt glands in our fish, it's another means of kind of isolating and storing salt for excretion later on. So very, very, very exciting and fun solution to the problem. Okay, so we'll cut the video here and we'll do one more as we move on. We'll talk about where urea, where uric acid, where ammonia, where all these things are kind of put on, and talk a little bit here as we finish that out as a nice transitional segue into terrestrial organisms.